Imagine trying to repair the wiring of something incredibly complex, like the hyperdrive on the Millennium Falcon after it's been totally fried. It's not just, a loose wire. The pathways themselves are fundamentally broken. And that rebuilding task, that seemingly impossible job, that's pretty much the challenge facing neuroscientists right now. We're talking about things like stroke, Alzheimer's, Parkinson's, spinal cord injuries. These are the biggest cause of disability globally and the second biggest cause of death.
Hmm.
And, well, the tough part is our current treatments, they mostly just manage symptoms. They might slow things down, but they don't actually restore what's been lost.
Exactly. The core issue really is that your central nervous system, your brain, your spinal cord, doesn't really regenerate much on its own. So when those key neural circuits are damaged or destroyed, traditionally they're just gone.
Right.
We need something new, a tool that pushes past just managing symptoms. We need something that aims for actual tissue restoration.
Okay, and that tool, that's what we're diving into today, right? This idea of 3D bioprinting trying to, well, bridge this huge gap in regenerative medicine.
That's the plan. Our mission here is to unpack some really key research, mapping out how this goes from, a concept in the lab towards something real. We've been looking closely at a 2025 perspective article in Advanced Materials. It's called 3D Printing for Neural Repair, Bridging the Gap in Regenerative Medicine. The authors are Mitchell St. Clair Glover, Zillian Yu, and Mirella Dettori, all from the University of Wollongong. And they really lay out a kind of blueprint for rebuilding parts of the nervous system.
Okay, let's get into it. When we say 3D bioprinting here, we're not talking about like printing plastic trinkets. This is living stuff. How does that even work for nerve tissue?
Yeah, it's basically the next level of additive manufacturing, but for biology.
Yeah.
So instead of plastic or metal, you're precisely depositing living cells. These cells are usually suspended in a special gel-like stuff called a bioink. And you use highly precise printers to lay this down, layer by tiny layer, building up a 3D structure that's designed to mimic how cells are organized in actual native tissue.
Wow. So it's like being an architect, but for microscopic living structures.
Pretty much. And the research we looked at highlights three main ways, three pillars, you could say, that this tech is being applied right now for the nervous system. Okay, so first they're creating really detailed in vitro models. Think complex tissues in a dish, used for studying diseases or screening new drugs. Second, there are cellular scaffolds. These are structures they implant without cells initially. They act like guides, encouraging the body's own repair cells to grow in the right direction.
Okay, like a trellis for nerves. And the third pillar sounds like the most ambitious one.
It definitely is. The third is cell-laden constructs. Here you're actually printing structures with living cells already included. The goal is to implant this living functional tissue to actually replace or reconstruct the parts of the neural circuits that are damaged. A real graft replacement.
That framework makes sense. Let's dig into pillar one first, those better lab models. We used to rely on, cells just flat on a petri dish, 2D cultures, but the brain is like the ultimate 3D structure. Studying it in 2D seems, like trying to understand a city by looking at a flat map. You miss all the height and connections.
Oh, the difference is huge. Real tissue has this complex cytoarchitecture, specific arrangements of different cell types, neurons, glia, all layered precisely. Plus, there's the extracellular matrix, the ECM, which provides physical support and chemical signals. 3D bioprinted models can replicate that spatial complexity way better.
And the paper shows this actually works better, right? They mentioned an Alzheimer's disease example.
Yes, exactly. They talked about bioprinted models using human neural stem cells engineered with Alzheimer's-related mutations. Compared to standard 2D cultures, these 3D models showed more aggregation of those problematic amyloid beta proteins and changes in tau gene expression that are much closer to what you see in the actual disease.
So why is that 3D structure so important for getting the disease model right?
Because the physical environment massively influences how cells behave. The 3D arrangement, the specific stiffness of the bioink, it's closer to the real brain. This pushes the cells to act out the disease processes more realistically than they would if they were just flattened on plastic. It makes the model a much better predictor. And here's something really interesting, kind of a wow factor. You might know about brain organoids, these amazing little self-organizing mini brains. While organoids are super sophisticated, their self-assembly means they can be quite variable. You don't get precise control over the structure. Bioprinting, though, lets you define the exact geometry. That means more consistency, more reproducibility, which is critical for things like reliable drug testing.
Okay, moving to pillar two. Those acellular scaffolds, the guides for repair. Let's talk peripheral nerves first, like in your arm or leg. if there's a big gap, say more than a centimeter or two.
Right, the nerve often just can't bridge that gap on its own. It fails to heal properly. So we need something to help it. And these bioprinted nerve guidance conduits, or NGCs, are looking really promising as alternatives to nerve grafts, which have downsides. Bioprinting lets you customize the conduit's internal structure, which turns out to be really important. Zhu and colleagues, for instance, used a fast 3D printing method to make personalized conduits. They tested them in rats with sciatic nerve injuries and found they supported recovery of both sensory and motor function. Pretty impressive.
And it's not just about the physical shape, is it? You mentioned loading them up with things.
Exactly. You can functionalize them. You can embed neurotrophic factors. These are chemicals that nerves love, like NGF or GDNF. They encourage survival and growth. So the scaffold provides the highway, and the factors are like signposts and fuel along the way, really boosting the body's natural repair efforts.
Right. Now, scaling that up to the central nervous system, like the spinal cord, that's a whole different level of difficulty, especially with the glial scar tissue forming.
It is much harder. In spinal cord injury, or SCI, that scar tissue actively blocks nerve regrowth. So here, cellular scaffolds can act as localized delivery systems. Chen and colleagues, for example, made a scaffold with immobilized FGF, that's fibroblast growth factor, another important signal. Crucially, they matched the scaffold's softness to the native spinal cord. When implanted, it didn't just guide, it delivered FGF right where needed to help reduce that inhibitory scarring, and they saw better motor function in their animal models.
Okay, which leads us perfectly into pillar 3, the cell-laden constructs. This is the really ambitious goal-printing living tissue replacements. For really severe damage where the body just has no hope of repairing itself, this is where we need to go, right? Actually integrating new functional tissue. The paper mentions some landmark work by Koffler and colleagues back in 2019, using scaffolds with neural stem cells for spinal cord injury.
Yeah, that was a big one. They implanted these bioprinted scaffolds, already loaded with neural stem cells, into rats with completely severed spinal cords. And the results were remarkable. The implanted cells survived, their axons grew across the injury site, they formed new connections, actual synapses, with the host neurons, and importantly, the animals showed significant improvement in their ability to move. That's not just managing damage, that's functional restoration.
And this ties into personalized medicine too, doesn't it? Using the patient's own cells.
Absolutely. Jin and colleagues demonstrated this using patient-derived iPSCs, induced pluripotent stem cells. They made personalized cortical grafts. The huge advantage is you can use the patient's own reprogrammed cells, so there's no risk of immune rejection. And their bioprinted grafts integrated structurally and functionally into the host brain tissue. It's a glimpse of truly personalized neural repair. And it's not just about the brain and spinal cord. The authors stress a really vital next step. We need to figure out how to integrate nerves, the peripheral nervous system, into other bioprinted organs. Think about engineered skin or muscle or a bladder. If you print a muscle but it doesn't have nerves to make it contract or feel, it's not very useful, is it? Right now, that lack of innervation is a major bottleneck limiting the function of most engineered tissues.
Okay, so the potential is massive, but the article is also clear printing neural tissue is incredibly hard. One of the toughest challenges in regenerative medicine. Let's talk about those big 3 hurdles they identified.
Right, the first one is just the sheer complexity. Biological complexity. The nervous system has so many different types of neurons, different types of glial support cells, and they need to be in the right places, in the right proportions, connected in very specific ways. Think about the neocortex, the outer layer of the brain. It has 6 distinct layers, each with different cell types and functions. Replicating that precise architecture with printing, that's extremely challenging.
Okay, complexity. What's the second hurdle? Especially for bigger repairs.
Vascularization. Getting blood supply, basically. Any tissue thicker than, say, a couple of 100 micrometers needs blood vessels to deliver oxygen and nutrients and remove waste. If you implant a chunk of tissue without a built-in blood supply, the cells in the middle will just die. And relying on the body to grow blood vessels in the implant that process, spontaneous vascular infiltration, is way too slow. We're talking less than 10 micrometers a day. The cells would starve long before the vessels reach them.
So you can't just print the cells and hope for the best. You need to engineer the plumbing too.
Exactly. Researchers are working on techniques like coaxial bioprinting, where you print a tube within a tube, or using sacrificial materials. The idea is to print channels within the construct that can act like preformed blood vessels, giving the cells immediate access to what they need once implanted.
Makes sense. And the third hurdle they mention is fascinating, the stiffness problem, how soft or hard the material is.
Yes, the biomaterial constraints, particularly mechanical properties. Neural tissue is incredibly sensitive to the physical forces around it, its stiffness or softness. And there's a huge range. Brain tissue is incredibly soft, thing like soft tofu, maybe zero point 0.1 to 2 kilopascals. But peripheral nerves, much, much stiffer. They'd be up to 5 megapascals. That's thousands of times stiffer.
Wow, I had no idea the difference was that big. Why does that matter so much?
Because mechanical cues tell cells what to do, especially stem cells. Neural stem cells, for example, tend to differentiate into neurons better on softer substrates, closer to brain stiffness. If you implant a scaffold that's too stiff into the brain or too soft for a peripheral nerve repair, you could actually prevent the cells from developing correctly. You could hinder the whole repair process just by getting the mechanics wrong.
So the bioink itself has to be this perfect sweet spot, soft or stiff enough to match the target tissue, but also printable and obviously safe for the cells.
That's the balancing act. And even if you nail the complexity, the vascularization, and the stiffness, there's still one more crucial step.
Which is?
Functional validation. Proving that the printed tissue actually works like real neural tissue. Does it conduct electrical signals properly? Do the neurons fire and communicate?
Right. How do you even measure that inside a dense 3D block of cells?
It's tough. Standard tools like multi-electrode arrays, MEAs, that record electrical activity, or calcium imaging that watches firing patterns, they often struggle with the thickness and opacity of 3D constructs. You can't easily see or record deep inside. So the field needs new tools. People are developing things like 3D integrated electrodes or flexible, stretchable electronics that can be embedded within the tissue or conform to it to really measure the electrical function non-invasively and confirm that it's not just looking right, but actually working right.
Okay, so we've got complexity, plumbing, stiffness, and proving it works. Those are some major challenges. Given all that, why are the researchers still so focused on bioprinting? What's the unique advantage?
The killer advantage, the reason everyone's pursuing this despite the difficulties, is spatial control. Unparalleled spatial control, what the authors call creating bespoke architectures. Bioprinting is unique in its ability to let scientists decide exactly where each type of cell goes, how the scaffold material is structured, and even embed other functions like sensors or drug delivery all at the same time.
Ah, so that controls how they can tackle that complexity barrier we talked about earlier by literally designing the tissue layer by layer.
Precisely. It allows them to try and replicate those 6 cortical layers, for example, or to place specific types of neurons, like dopamine producers for Parkinson's models, exactly where they need to be. And embedding sensors is powerful too. They mention work by Lai and colleagues who put tiny zinc oxide particles into a bioink. These acted as built-in optical sensors to measure dopamine release in real time within the printed tissue.
That's incredible. Merging material science, engineering, and neuroscience right there in the printer.
Yeah.
Okay, before we wrap up, we should probably touch on the hurdles that aren't purely scientific, the ethics and regulations.
Definitely. Ethically, there's always discussion around the cell source. Human embryonic stem cells, HESCs, have ethical concerns for some, which is why iPSCs using the patient's own reprogrammed adult cells are becoming much preferred. They avoid the HESC debate and offer that personalized, immune-matched advantage.
And then there's that sci-fi question. As these models get more complex, could they become conscious?
Yeah, the sentience question does come up, especially with advanced brain organoids. It's a valid philosophical discussion to have as the technology progresses. But realistically, current bioprinted constructs and even organoids are nowhere near the scale, complexity, or integration needed for anything like genuine thought or self-awareness. It's more of a future consideration than a present reality.
And getting these things approved for human use sounds like a regulatory headache with all the different components, cells, materials, growth factors.
It is complex, absolutely. Regulators are dealing with combination products, which need new frameworks. But the encouraging part is progress is being made. That 2025 review we mentioned, it identified up to 11 registered clinical trials already involving bioprinting for various applications. So it shows the path from bench to bedside is starting to be paved, slowly but surely.
This has been a really fascinating look at a field that feels like it's right on the edge of science fiction becoming reality. If you, listening, take away just 3 key things from our chat today, let them be these. First, 3D bioprinting is aiming to completely change the game in neurorepair. It's about moving beyond just managing symptoms towards actively restoring function in the brain and nerves.
Second, this isn't just one approach. The technology is moving along three main tracks: building more accurate 3D lab models to understand disease and test drugs, creating structural scaffolds to help the body heal itself, and developing living cell-laden grafts for actual tissue replacement.
And finally, getting there means tackling those huge technical hurdles head on, mastering the cellular complexity, figuring out the vascularization or plumbing, and developing ways for functional validation proving the printed tissue actually works.
Yeah, and the big message from the researchers is that there's no single magic bullet. Success will require custom solutions, bespoke, tailored therapies designed for specific injuries and potentially even individual patients. It's all about combining the best of stem cells, material science, and neuroengineering.
Okay, and here's something to chew on as we finish. We talked about how sensitive neural tissue is to stiffness, that super soft brain, versus those much stiffer peripheral nerves. So imagine if scientists get so good at this that they can print a replacement spinal cord section that doesn't just have the right cells, but perfectly matches the exact mechanical feel, the precise softness and elasticity of the original healthy cord. What unexpected levels of functional recovery might that kind of physical precision unlock? Something to think about. We'll definitely be keeping an eye on this field.